There are a couple differences between a virus that infects an animal cell and a virus that infects a bacteria. So the first difference right off the bat is the name itself. We don't have a specific name for animal viruses, but for a virus that infects a bacteria, we call it a bacteriophage. But the general outline of the virus cycle remains the same. We have the first step. Obviously we need the virus to attach the receptor on the host cell, and we need the insertion of the genetic information inside the host cell. And then we have to either go in the lysogenic cycle, the lytic cycle, or if you're talking about animal cells, we have another type of cycle called the productive cycle, which is very similar to the lysogenic cycle in the sense that it's incorporating with the entire genome, but the end goal is slightly different in the sense that it's not going to enter the lytic cycle, and that it's just going to cause the virus to leave through exocytosis. And if an exocytosis is just when we have, imagine that the virus is leaving, it's kind of passing through the membrane of the host cell, and as it's passing through, it's going to steal some phospholipid bilayer of the host cell, and this will cause the formation of an envelope, and we'll talk about that in a second. So we have already talked that the entire process is similar in terms of the process of infection, the use of the host cell machinery, replication, synthesis of the capsid, assembly, and the entire process is very similar. So let's get into animal viruses and talk about it in more detail. So animal viruses, the proteins on the surface are specific to the virus. So, and let's give you an example real fast what I mean by that. So the HIV over here, we know this very commonly. The HIV it has a protein on it, and you can see there this purple protein over here called the Gp120 protein. Now the Gp120 protein is specific to T cell membranes, or the CD4. And because it's specific to the CD4, it allows the attachment of the HIV to the host cell, which can then cause the insertion of the genetic information, and then eventually, if you have the genetic information inside the host cell, it's infected. But the entire thing that I'm trying to highlight here is that infection cannot happen if the receptors are not compatible. There's no way of the viral genome getting inside the host cell. It's just not going to impact the organism at all. And that's the highlight of this video over here. So receptors play a very important role in human function. So yeah, now you might be questioning yourself, why the heck do we have receptors in the first place? Why can't we just get rid of these receptors so we don't get affected by viruses in the first place? So smart. But the problem is that these receptors are not just there for viruses, obviously. They're playing normal roles in human function in terms of the HIV. And the T cell, it's responsible for a couple of things, and without it we won't be able to survive. So we need the T cells. We can't just destroy the receptors. It's going to cause other metabolic problems. We can't do that. So administering a soluble form of CD4, do you think it will help control the virus, and why? The answer to this question should be yes right off the bat, because soluble CD4 will bind to the Gp120 of the HIV. And of course, these methods are used if we know that there are some methods of controlling viruses when you have HIV. It really reduces the rate of you getting, or God forbid, not you, but whoever has HIV, it slows down the process of getting AIDS or developing AIDS. And we know this process is slowed down by a huge factor. But if you have HIV, but it's not cured, you will have the virus in your body, and there will be several outbreaks at times, but you'll be consuming these pharmaceutical drugs or whatever you're consuming. In this example, if you're consuming a soluble form of CD4, it will be binding to the virus. And now the receptor is binded, it can go and infect the host cell. So it's slowing down the process massively. Mutations on the cell surface with receptors means immunity? Question mark. Well, think about it again. If the virus doesn't have the ability to attach onto the host cell, it's obviously going to not cause the infection. So yes, receptors cannot attach, so therefore there's immunity. And there were a couple of cases in terms of HIV. There were some kids found in Africa who were tested positive for HIV when they had a blood test, but they showed no they showed no they reflected no they reflected none of the characteristics of HIV. So they found out their receptors were mutated, but this is not a normal or very normal thing to watch for. It doesn't happen normally inside. It doesn't happen normally, what I'm trying to say. It's uncommon because of two reasons. The first reason, viruses rapidly change and can evolve to resist. And we know that it's not DNA, and viruses are going to have numerous mutations, therefore, because if we have DNA, we have something called exonuclease activity, which is basically proofreading, making sure that there are no problems or no mutations inside it. We might have made a mistake. Let's go back and check it. So it's going through from a 3' to a 5' in terms of the strand and checking if there are any problems on the strands. But RNA doesn't have that. So if it's the RNAs, there are going to be numerous mistakes made. So because of the mutations, there's so much. The virus itself is replicating so fast that we even have a hard time catching up. And if we even were able to find a solution to the virus, by the time we find a solution to it, it's just going to be a whole new different characteristics on the virus because it's replicated so fast and made so many mistakes. And that's another issue we got to deal about with viruses. So now HIV. So the first thing we're going to talk about is the HIV. So Gp protein specific to the treatment. We talked about this right now. Insertion of viral genome through endocytosis. Plasma membrane of the virus integrates with the host. And we have one other difference is the insertion of viral genomes through endocytosis. Endocytosis is a process when we have viruses that are basically penetrating through the host cell. It's going to be kind of enveloped inside the phospholipid bilayer, and this will cause the entire genetic information to enter the host cell. This process is called endocytosis, and this can happen with really anything. And just refers to the fact that there is a envelope or wrapped. Well, the phospholipid bilayer kind of wraps around, engulfs the whatever is coming inside the host, and it allows the passage of it inside the host cell. Now, I already talked about there are 3 cycles we have in animal cells. We know the Lytic cycle. Its end goal is to just produce a lot of viruses, and in bacteria it's 100 progeny for estimate 100 progeny. So just a lot of viruses produced, kills a host cell. Lysogenic cycle, dormant for a while, but eventually enters a lytic cycle, kills the host cell. Productive cycle is like the lysogenic cycle in a sense, because it integrates with the host cell, but then it causes exocytosis, and this will cause the host cell to be alive throughout the entire thing. And I've said it right now over here. So animal lysogenic cycle. Let's compare this with in terms of the vocab you need to use. So if you see the word provirus, that refers to the animal virus with a host. An animal virus integrated with a host genome. That's a provirus. A prophage, on the other hand, is a bacterial genome with a host genome. That's important to memorize. And herpes simplex 1 is an example of an animal lysogenic virus. And that's because you might notice that if somebody has herpes, then they will generally have these outbreaks from time to time when they reflect the characteristics of the virus. There's some times when it's really not that bad, and that's because it's a lysogenic cycle. So when they're put under stress, let's say they have a school exam coming up, they have a very bad day, they have some ice cream, they just have a bad day, you had a bad date, they have a stressful day. This is going to put stress in the cell, and during time of stress there will be expression of the virus, and then they reflect the characteristics. So that's the concept behind the herpes simplex. So that's why it behaves in a certain manner. Now, hopefully you have a better understanding of the differences between animal viruses and bacteriophages, we want to call it. So hopefully this video makes sense. Thank you.